Yes, thank you very much for this very kind invitation and for the introduction. So my talk will be about multivariate circulating immune signatures for diagnosis, prediction, target identification, but also for mechanistic insights in thromboinflammation and immunothrombosis. Now, a few words about myself. I did my medical studies in Munich and was subsequently in a DFG-funded clinician scientist GEROC position. I did my MD thesis in 2020, and then I joined a structured clinician scientist program as a cardiologist in training, put in parallel research time for 3 years. Since this year, I've been awarded a DZK, so a German Center for Cardiovascular Research junior research group position, also in Munich at the cardiology department. Now, what did my research, what did I focus on in the last let's say decade? I focused on the role of the immune system in the development of cardiovascular disease. On the one hand, on the circulating immune response and how the circulating immune response interact with the coagulation system in different kinds of septic, but also sterile thrombotic disease. But I also focused on tissue resident immune cells and on how the vascular vessel-associated innate immune cells are actively maintained and how they modulate the development of atherosclerotic, but also atherothrombotic and venous thrombotic cardiovascular disease. Now, in the past, let's say century, the immune system has been identified as a central gatekeeper of the development of cardiovascular disease. Different kinds of cardiovascular risk factors, particularly LDL cholesterol, diabetes, smoking, etc., have been associated through mobilization and priming of different kinds of circulating and immune and local immune cells to foster the development of cardiovascular disease. For example, the detrimental remodeling after myocardial infarction, but also the accelerated progression of atherosclerosis in the vascular wall. Now, most of these studies here, you can exemplarily see the recruitment of innate immune cells to the myocardium, for example. Most of these studies and most of these mechanistic insights throughout the last decades have actually used mouse models or rodent models to identify these mechanisms and provide detailed insights. However, whether and how this is transferable to the human system currently remains unclear. So the immune response in human immune coagulation interplay, in human thrombotic disease, and in human ischemia reperfusion remains vague. So the key point that we first focused on, the key question was to unravel the systemic immune landscape in a arterial thrombosis disease, in an arterial thrombotic disease, in this case, one of the most important diseases, myocardial infarction. So we wanted to understand whether and how the circulating immune cells in humans respond to myocardial ischemia. And here we focused on patients with acute or chronic coronary artery disease. Now, we included two different cohorts, one in Munich with patients with acute coronary syndromes. All of them had an ST elevation myocardial infarction. We include patients with chronic coronary artery disease, stable coronary artery disease, and patients with a rule out of coronary artery disease. And we included several time points of ACS. So the acute time point then 14 hours, 60 hours, and before discharge, so 5 to 8 days after the acute event. We also included a second validation cohort from patients from Groningen from a second study. So what did we analyze? We focused mainly on the circulating immune system. So we included mainly whole blood, peripheral blood mononuclear cells, which we isolated, blood plasma, as well as neutrophils. And the assays that we performed, obviously, we included clinical values from these patients, so mainly troponin, CK, CRP, etc. We included single-cell RNA sequencing, flow cytometry, and different kinds of protein-based assays, as well as prime-seq, which is a kind of bulk RNA sequencing of neutrophils. What do we subsequently do? So when you perform multi-omic measures, you obviously get a huge set of data, which is particularly difficult to interpret. So what we did in collaboration with Matthias Heinig from the Helmholtz group was we integrated these multi-omic data sets to one joint integrative data set, and then we performed a data-driven dimensionality reduction called MOFA, which is multi-omic factor analysis. So what does factor analysis do? Factor analysis identifies distinct variables that correlate with each other among different patients. So let's say, for example, CRP, leukocytes, interleukin-6, interleukin-1 are upregulated in one patient because, let's say, the patient has some kind of an infection, and they are normal in another patient, then factor analysis would identify these variables to be correlative and would stack them into one factor. So these correlative variables would be summarized into one factor. So the good thing about factor analysis is it reduces the dimensionality of multiple variables and identifies key axes of variance in the big data set. So first of all, what did we identify? When we just look at the single cell RNA-seq and the cell clusters that we identified. So we identified most clusters that we know from circulating immune cells, and we could see that mainly T cells, but also distinct monocyte clusters change quantitatively over the course of myocardial infarction. But this doesn't tell us too much. I think what was particularly interesting, and the reason why we did this study was to understand these immune signatures, these factors that we might identify. So what kinds of factors did MOFA identify? Well, we had a total of 20 different factors, and this heat map now shows which factor explains the variance in which data set. So these are all the data sets that have been included in the factor analysis. And as you can appreciate, factor 2 seems to be the factor that most strongly, or let's say best, explains the interpatient variance, explains the variance within the clinical values. So troponin, CK, CKMB, etc. This is for us as doctors a particularly interesting factor because obviously we are interested in clinical values. So we dived into factor 2, and we asked ourselves, okay, when factor 2 explains the clinical variables so well, what is factor 2 actually built of? So what makes up factor 2? What defines factor 2? And as you can see here, factor 2 is actually surprisingly mainly built of immune cell variables, single cell immune cell variables. So mainly CD8 positive T cell variables, CD4 T cell variables, and monocyte variables. And the clinical values, they do also influence the factor, but at least not as much as T cells. So here you can just see the top 1% of features. Anything that's above the 1% would not be included here. But importantly and surprisingly, even though factor 2 explains the variance in the clinical values very much, it is not mainly built by the clinical values. So it's defined by immune cell signatures, because which is interesting, because it seems to explain the disease cause and the clinical values, but it seems to be mainly derived from single cell clusters. So you would expect that factor 2, if it explains the clinical values, behaves also similar to the clinical values, which it does. When you look at the development of factor 2, you can see that at early time points of myocardial infarction, you see particularly high expression of this factor, which drops to normal levels in both cohorts upon time. And due to the fact that the factor is mainly built by immune cell signatures, but very well explains the disease cause, we named the factor integrative ACS ischemia because it summarizes the immune response to ACS ischemia, behaves as you would expect from biomarkers to ACS, but is mainly built by immunological variables. Now, what specific pathways characterize this factor? What specific pathways characterize the immune landscape in ACS? So what is factor 2 specifically made up of? When we perform a pathway analysis, we identify different cytokines, mainly interleukin-6 signaling, as I said, mainly in T cells and monocyte variables, but also in circulating plasma data sets. So this is not unraveled here, but this is just a joint pathway analysis. And these cytokines are particularly prominent in defining this factor 2. Now, we hypothesized when cytokine signaling across different immune cell subsets defines integrative ACS ischemia, we hypothesized that when we add plasma from patients with acute coronary syndromes to healthy immune cells, in this case monocytes, we should be able to recapitulate a kind of an activated or changed monocyte phenotype, which indeed we could. We did see an activated monocyte phenotype as derived by characterized by flow cytometry, and also using functional assays, we could observe an increased capacity, an increased efferocytotic capacity of monocytes upon addition of ACS plasma to healthy monocytes. Now, when this shift of monocytes based on adding ACS plasma to monocytes is truly mediated by cytokine signaling, then inhibiting interleukin-6, for example, should reverse this shift in monocyte activation. And this is what we tried. So we added, again, ACS plasma, but this time either with an anti-IL-6 antibody inhibiting IL-6 signaling or with an ICE-type antibody. And we could at least partially reverse this shift of monocytes upon addition of ACS plasma to healthy monocytes. So summarizing this project, we profile the longitudinal immune response to myocardial ischemia and define the disease state, which we term integrative ACS ischemia. I didn't show this data due to time reasons, but we also identify a factor which we term integrative CCS, which strongly associates with the presence of coronary artery disease, stable coronary artery disease. And we identify one factor that associates with a positive functional outcome upon acute coronary syndromes, which we term integrative ACS repair, because it also associates with a late stage repair signature, which associates with a positive development of cardiac function over time. From a pathophysiological perspective, we identify plasma driven factors that mainly mediate the cellular shifts during acute coronary syndromes. Now, this provides a first map of the systemic response to myocardial ischemia in humans, strictly linking clinical features, a clinical disease cause with the immunological signatures, and integrating the clinical side with the immunological side during myocardial ischemia. And this provides a resource that might provide novel therapeutic, but also diagnostic candidates that might serve as for future basic or translational studies. Now, obviously, as we all know, correlation does not equal causation. So when we analyze the circulating immune response to a disease that involves arterial thrombosis, that does not necessarily allow us to tell what is going on within the thrombus itself or within the myocardium itself. So the next question we asked was, what does the local immune response within the thrombotic occlusion, so within the thrombus in humans, look like? The concept of thromboinflammation, so the concept of the mainly innate immune system interacting with platelets or the plasmatic coagulation system, has been on the rise throughout the last decades. And the concept of thromboinflammation has mainly been thought to be prothrombotic, detrimental, and something worth to inhibit to actively prevent the development of thrombosis. However, again, most of the studies that have been performed in rodents, and they are only mainly histological studies on human thrombi. So we asked whether immune coagulation interplay might also hold some pro-resolving protective effects in thrombosis. And particularly, we focused on humans because, as I said, this is still highly understudied, mainly by novel state-of-the-art methods. So we again chose a cohort study design where we included thrombi that had been retrieved by thrombectomy from humans. And we performed single-cell RNA sequencing again and prime-seq histological assays, flow cytometry, and we performed reverse translation of the key findings that we had in humans to a mouse model and an in vitro model, allowing mechanistical validation of the key findings that we had in the human disease model, in the human thrombi that we had analyzed. So what does the immune landscape of thrombosis in humans look like? So immunothrombosis at single cell resolution. In humans, in human thrombi, the most frequent immune cell subsets are actually monocytes and neutrophils. And this is something that is, to be honest, highly has been shown multiple times in the past. So this is not really new, to be honest. Monocytes and neutrophils have histologically been described several times to be highly frequent in human thrombi. But what we can see here, that in addition to the fact that they are highly frequent, they are also the immune cell subsets that most strongly and most prominently shift phenotypically. So these monocytes within thrombi actually form completely new subsets that are not present in blood. And this is very similar also in a mouse model of thrombosis. Now, when we focus on neutrophils and recluster neutrophils, we, as I said, observe a very strong shift of neutrophils with the development of a VEGF-A high neutrophil subset in thrombi. And when we want to further understand what defines these neutrophils, we can observe that they express particularly high levels of VEGF-A, VEGF-B, thrombomodulin, urokinase receptor, etc. So these are actually genes that are associated with a pro-resolving phenotype. And also in a bulk-seq approach, we can validate that urokinase is strongly upregulated, which is surprising because urokinase is actually an agent that is administered by neurologists during an acute ischemic stroke, which is a plasminogen activator that allows thrombolysis, so that allows a quick resolution of thrombosis. It's a therapeutic agent. So neutrophils themselves, which are actually known to be prothrombotic, seem within an already established thrombus, seem to express high levels of an agent that is, or a gene that is known to be thrombus resolving, so known to be highly let's say, therapeutic in this setting. So it seems to be that the neutrophils within thrombi seem to kind of want to free themselves or kind of resolve the thrombus again. Now, what other genes do they express? They express high levels of hypoxia-related genes, HIF1 alpha, HIF1 alpha AS3, for example. And when we perform something called gene network correlation analysis, which just stacks genes that seem to correlate in between cells into modules, we can observe that HIF1 alpha, that there is a module where HIF1 alpha is co-expressed with these pro-resolving genes such as VEGF-A and thrombomodulin. So we hypothesized that maybe hypoxia and hypoxia-related signaling might be the key inductor of this neutrophil, of this pro-resolving neutrophil phenotype that develops within established thrombi. And indeed, when we knocked out HIF1 alpha in mice, specifically in myeloid leukocytes, we observed, first of all, an exacerbation of thrombus weight in an inferior vena cava reduction, flow reduction model. And this was accompanied by a significant reduction in these pro-resolving neutrophils. Vice versa, when we add roxadustat, which is a HIF1 alpha stabilizer, we can in vitro reduce thrombus weight over time. So, summarizing this, HIF1 alpha signaling in neutrophils seem to induce a pro-resolving neutrophil phenotype, surprisingly, within thrombi. So what about monocytes? What are the monocyte phenotypes within thrombosis? So monocytes also seem to undergo a very strong, very prominent shift compared to blood in thrombi. They seem to build a completely novel immune subsets within thrombi, which are not present in the blood. And they are also defined by high expression levels of urokinase receptor, so different pro-resolving genes. They express high levels of CD16, which is why we term them non-classical, like thrombus monocytes. But what they also express is high levels of neutrophil chemoattractants, CXCL8, for example, MIF, CXCL1. And this is something that was to us surprising, because why would monocytes within thrombi express high levels of neutrophil chemoattractants? The thrombus is already packed with neutrophils. Why would we require even higher levels of neutrophil chemoattractants? So we hypothesized that this might have some evolutionary or some functional relevance. We hypothesized that maybe there might be a continuous influx of neutrophils into these thrombi. So maybe the neutrophils that we see in the thrombus might not be the same neutrophils that had been in the thrombus during the development, but have subsequently influxed into the thrombus. So to follow up on that, we employed a fate mapping model where we labeled different surges of neutrophils with different colors, green and red, and we could observe indeed different subsets of neutrophils present in the tissue. So green neutrophils, first generation neutrophils, and red neutrophils, which have infiltrated the tissue after 48 hours after the thrombus had already evolved. And interestingly, most of the neutrophils that we could observe in the thrombus were actually second generation neutrophils, confirming that there is a continuous influx of neutrophils after the thrombus has already been established. So the neutrophils that we observe in established thrombi are not the same neutrophils that have originally been involved in thrombus induction and the development of thrombosis. So is this influx of neutrophils mediated by non-classical monocytes? As I've said, they express high levels of neutrophil chemoattractants. When we delete non-classical, or when we deplete non-classical monocytes by deleting NR4A1, which is the transcription factor that allows the non-classical monocytes to develop, and then we indeed can deplete these non-classical monocytes. So this is a technical confirmation. And this results in a reduction of thrombus neutrophil content, which is associated with an exacerbation of thrombus weight. So summarizing this, we using this cohort study of human thrombi and employing a single cell based atlas of human thrombosis, with subsequent reverse translation to mouse models and in vitro models, we observe monocytes within thrombi, non-classical like monocytes, to continuously express high levels of neutrophil chemoattractants, allowing neutrophils to continuously influx into thrombi, developing a hypoxia-related pro-resolving phenotype, which continuously reduces thrombocytes and resolves thrombosis. The interference with this axis resulting in exacerbation of thrombosis in mice. And with that, I would like to end, and I'm happy to take questions.